Hello, my name is Thomas Elliott, and I'd like to give you a very brief and light-hearted overview entitled Embryologists versus Robots. There are some concerns that as robots become more intelligent, not only will they take over some of the tasks that we perform, but they may even take over our jobs. They could even take over the world. Interestingly, in a 2017 survey, 65% of respondents felt that jobs would be lost in the future to automation. However, they also felt that their job would be safe. We do have some historical information about what can happen when automation enters the working environment. Back in the early 80s, when ATMs were first introduced, there were fears that machines would make human bank tellers obsolete. However, what actually happened was because of cost savings, many more small banks were opened and the total employment actually went up. This may be a model for what is to happen in IVF as automation becomes more common. So instead of a few centralized locations, there will be many more smaller clinics spread throughout, each staffed with a smaller number of embryologists. We already have some automated IVF processes, either in use or perhaps in the near future, including sperm separation devices, automated vitrification, and robotic cryotanks. There are also some outspoken people who believe the embryology of the future will be very different. Dr. Santiago Munet, the COO of Over Your Life, a company which is presenting today, thinks that the embryology job of the near future will either be a low-paid job of feeding samples into a machine or perhaps a high-paid job programming these machines. We already have cars that can drive themselves, robots that can cook for us, perform complex surgeries. We even have robots that can replace relationships. Is it too far-fetched to think that robots can't replace an embryologist? I'm very excited to hear these next few talks and find out, in 30 years, will there still be human embryologists?
Well, thank you, Thomas. Thank you very much for that video. Welcome to the 7th session of the International IVF Initiative. I'm honored and excited to be co-hosting this session with Jacques Cohen. For videos of previous sessions, please go to ivfmeeting.com. You'll also find the information about upcoming sessions there on the same page. Our continued goal is to support IVF community during the COVID-19 pandemic and provide educational opportunities while bringing our community together. It's great seeing so many people from all over the world, reproductive biologists, reaching out and taking part in these sessions. Thanks for logging in, listening, and please keep those questions coming. My name is Giles Palmer and I'll be co-moderating this session with you. We've got some amazing programs today, planned for around about 80 minutes or so. The theme today is assisted reproduction, 2049. Will there still be human embryos? We have progressed far. Think of what's happened in the IVF lab in the last 20 years, or even in the last 10 years. But we still need to reduce human error and the variability of the results and make treatment available for all. As technologies converge and the boundaries of one discipline of science merge into another, it's a matter of time that more and more things will become robotic or automated. In fact, robotics is all around and you maybe just don't realize it. As this world that we live in now resembles a sci-fi movie, it's time for the iCube team to look into the future with talks about flexible robots, automated procedures, and even IVF labs in a box. We have a talented team of our own, our little robots who are working behind the scenes to pose your questions to our speakers. I encourage you all to use the Q&A section of the conference service and not the chat function. Some questions will be addressed live and some will be typed into the Q&A section. So from this grey annotator, I'd like to hand you over to our very own soft manipulator, Jacques Cohen, who will begin our discussion.
Well, thank you, Giles, for that great compliment. Before we introduce the panelists, I wonder if Thomas can show us some poll questions that attendees can answer. But, you know, panelists can answer this too. So while we wait for the poll results to come in, it's my great honor to introduce you to our 4 speakers today, who are well versed in this area of research and development. The 4 speakers come from France, Israel, Spain, and the UK. And they are talking to us from those locations now. Professor Saaj is the Global Chair in Robotic Engineering at the University of Lincoln, the UK. She's Head of Research in Industrial Digitalization and System Intelligence. Mini, I'm going to introduce all 4 speakers first. Previously, she was the Head of Robotics and Control Research Group at the Surrey Space Centre in the UK, so she's a space robotics engineer. She has made the leap to terrestrial application and is interested in a wide variety of clinical application. From France, Dr. Arnaud Reignier is a clinical embryologist, one of our very own at the Art Center of the University of Hospital of Nantes in France. He is also a member of the UMR 1064 research team in the main pluripotency and embryo development group. His main interest is that of embryology, time-lapse, pluripotency, and pre-implantation physiology. The third speaker will be Dr. Amir Arav. He is a doctor in veterinary medicine and has a PhD in neuroscience and bioengineering. He has dozens of patents and is published widely. And finally, Garcia Poyo is an industrial engineer and the Vice President of Operations of Overture Life, based in Madrid, Spain. She has been instrumental in developing to market products for polyoptics, Zeiss, and Eurospace. So we have 3 engineers and 1 embryologist. It's my great pleasure to introduce you to Professor Saaj from Lincoln University, who will talk about soft robotics and potential applications in assisted reproduction. Thank you, Mini.
Thank you, Jacques, for this introduction. I hope all of you can hear me. Hello, everyone. First of all, I would like to thank the International IVF Initiative for this invitation. In addition to sharing my research with the audience and other panelists, this is a great opportunity for me to also learn from other speakers as well as hear the feedback from the audience. So, audience, are you ready to listen to a space roboticist who is going to talk about robotics or soft robotics in the field of assisted reproduction? Well, before I move on, I have to share this copyright and disclaimer just to remind everyone. I would like to say a few words about my institute. I'm representing the University of Lincoln. We are based in the northeast of England in Lincolnshire, the birthplace of Sir Isaac Newton. University of Lincoln is spread across 3 campuses, and at University of Lincoln, we undertake high impact, industrially driven research that has a very positive impact on our society. Within the Lincoln Center for Autonomous Systems, which is a cross-disciplinary research group at university level, we undertake extensive research in the areas of mobile robotics and robotic manipulation for several applications. And myself, I focus more on space robotics and technology transfer to several terrestrial domains. And one of my interests is in medical robotics, and this is the reason why I'm here today to share my research experience working in the area of soft robotics and how this could actually revolutionize the field of assisted human reproduction. So let's take a glimpse at what is the current state of the art of robotic surgery. Well, we all know that the Da Vinci robot from Intuitive Surgical is nowadays very widely used for minimally invasive procedures. However, as you can see from this clip, these are huge, expensive robots. They are made of rigid arms, very much like a standard industrial arm. So it's quite hard to actually reach those inaccessible sites within the human body. Interestingly, the next generation of medical robot from CMS Surgical is a modular version, which is again a rigid manipulator. So the soft robotics community worldwide is actually working very hard to develop innovative, soft, flexible, modular robots, which are actually inspired by the nature. So it's a very interesting to see, for example, how an octopus can actually squeeze through very narrow openings. So similarly, if you're able to design flexible medical robots that can squeeze through narrow trocar ports or other openings in a human body, that will be a great success. Here, it's important to say that the soft robotic community worldwide have achieved a lot. And there are many examples of functional or working prototypes. Here you can see a few that is actually driven or inspired by Octopus at the top, which is a stiff lob robot, an EU-funded project. Similarly, there are several other examples of flexible soft robotic grippers, which are very widely used for not only medical applications, but also for the agri-food industry and other applications. So soft robots, these are very different to the rigid counterpart because they can actually maneuver into very confined regions, and they can be made biocompatible, which is really important. And also cost is something that drives everything. So we are looking at how we can develop soft robots that are less expensive compared to the rigid medical robots. And of course, they should provide better flexibility, maneuverability. So if you look at this clip, you can see that the stiff lob project, which was an EU-funded project, we were able to successfully demonstrate both using ex vivo testing as well as human cadaver testing that an octopus-inspired, pneumatically actuated robot, a modular robot with embedded sensors. These systems can be really great if we are able to move into the next phase of development and get the approval from the clinical community. So this is a great example of one of the cool robots, so soft robots, I must emphasize that. Similarly, we were also able to work on other soft robotic instruments, say for example, for gynecological procedures. So the soft robot named as Gendler, this is a good example of a flexible uterine manipulator that could eventually replace the conventional rigid uterine elevators. So we were able to look at the feasibility of this system, starting from the initial concept, going all the way through the design, prototyping, and undertaking ex vivo testing. And we were able to actually meet several clinical requirements set by our clinical partners. And this is an ongoing work, and you can find more details about the different aspects of design or the mechanism, the sensing, control, etc. In various publications from our team. So I'm happy to share the full papers if you're interested to read them. But let's move on to the field of assisted human reproduction. Although I'm focusing more on the human side, this is equally applicable to veterinary medicine. If you look at this cycle of human reproduction, thanks to my daughter for sketching this out, you can very well see that there are several areas where we can apply robotics, automation, and AI techniques. So for me as an engineer, one of the starting points is actually to look at how we can support doctors in terms of accessing the uterus more efficiently, accessing or identifying follicles that contains healthy oocytes, and also to make sure how we can actually support the doctors as well as patients in terms of easy retrieval of healthy eggs. At the same time, it's also important to make sure that we support the embryologist in terms of the whole vitrification process, which is automated partly these days. There are several companies who are driving this market. For me, one other important aspect is to see how we can actually help again the embryologist and the medical team with precise implantation of the embryo inside the uterus. So at present, if you look at the state of the art of fertility treatment, it is heavily done manually by the doctors or the embryologist. And there is very little work in the area of robotics or robotically collecting the egg. So for example, if you look at the literature, you can see that, yes, there have been some work done in this field, but even today, robots are not actually finding their way into the fertility clinic. And this is because what has been done is using the traditional rigid robots, and they rely heavily on ultrasound images. Both are equally not good. And therefore, the soft robotics community could potentially help embryologists or the fertility team by introducing flexible soft robotic instruments, which could actually assist doctors in vivo procedures, including identifying follicles that are healthy enough, identifying or retrieving of ovum. They can help embryologists with identification of ovum or sperm using high resolution 3D imaging and other AI techniques such as machine learning. Similarly, there is a lot of work done in the field of automating the vitrification process, but there is a long way to go in terms of, say, for example, using AI techniques for identifying healthy embryos. And this is important to ensure that we can minimize the degree of miscarriages. Similarly, we can use, or we want to use, soft robotic instruments for in vivo implantation of embryos. And if we can do this using high resolution image guided procedures rather than ultrasound, the rate of success will be much higher. So looking at the potential uses of soft robots in this assisted reproduction sector, we believe that at present, the demand versus service ratio is not great. And with introduction of robotics, automation, and AI techniques, there will be more and more opportunities, which means, there will be more clinics, there will be more opportunities for embryologists. Similarly, we will be able to offer better outcomes, of course, at a lower cost compared to the current instruments or current robotic platforms available. The extent of skilled staff required, or the embryologists required now, is high. But at the same time, there is a shortage of embryologists. So we can very well descale the skills of senior doctors or highly skilled embryologists needed to undertake all these procedures if we have robotic or AI solutions. So moving forward, at this particular moment, I would like to recall this popular saying by poet Alexander Pope. Well, hold on for a second. Is this actually acceptable, IVF errors? I believe no. So the question is, through automation or through the use of robotics and AI, we can very well improve the quality of service currently offered by embryologists. So I believe by 2049, we will be able to make these solutions available to clinics, which means we will be able to see more and more fertility clinics popping up, which means more opportunities for embryologists, and we will be able to provide better care for the patients. So I would like to emphasize that as an engineer, as a roboticist, I believe there is a huge potential for soft robots to change, to revolutionize the art of assisted reproduction. And IVF errors will no longer be the tip of the iceberg. So this means we will be able to see more and more happy parents and happy kids and a healthy society. So with this, I would like to take any questions from the audience as well as from the panelists.
Thank you very much, Professor Saaj. That's a great introduction. Just one question before we go to the next speaker. Then there's a lot of questions already noted here from the audience. But just one question while we transfer to the next speaker. From Peter Parker in the UK. what's the main challenge that is special for IVF in robotics? What is different from other fields that you have worked in that makes IVF different?
Well, IVF and application of robotics is very different to other sectors, because here we are actually dealing with very clean conditions, clean environments. It is not as extreme as a space robot or a nuclear robot, but looking more specifically into the IVF part or the fertility part, because we are dealing with ultra small eggs or sperms or embryos, there is a good potential that soft robots can be easily implemented there. Because one of the biggest challenges soft robots face is the limited load carrying capacity, which is not a big issue or no longer an issue for IVF. So I believe there is a great potential.
Great. But it's a matter of scale too. You're working with patients, which is a scale you're familiar with. And then there's, of course, the very small patients, spermatozoa, eggs, and embryos. And they're on a different scale. I don't know, Thomas, if you have the results of the survey, of the poll ready. Yeah, there they are. The first question was, do you think that robots, AI, automation will replace the majority of a reproductive scientist's task in 30 years? And it was a toss-up. Yes, the answer was 42% and no was 45%. And the rest said, I don't know. So that is interesting. The second question, how do you feel about robots, AI and automation coming to IVF? And 9% is not worried, or is worried, sorry, 9% is worried. 11% is not excited. 23% is neutral. Somewhat excited is 47%. And then 9% says, I can't wait. So the majority, slight majority here, sort of embraces the panelists and speakers and says, well, let's have it. Let's see what you have. So somewhat excited and very excited is 56%. So that's not a bad start. So they're not necessarily shooting you all down, okay? So having said that, it's my pleasure to introduce Dr. Arnaud Reignier, and he will talk about automated annotation of time-lapse. Welcome, Arnaud. Your microphone is muted. Thank you.
Okay. So, hello everyone. Thank you for the opportunity to speak today. And I'm a clinical embryologist in Nantes University Hospital. And we are going to speak about automated annotation of morphokinetics events with time-lapse analysis of embryo development. So first, I wanted to say that I have no conflicts of interest to declare for this presentation. And so we all know that time-lapse systems give us the possibility to study all the events of embryo development without disturbing the culture conditions. So normally, giving optimal culture conditions and being able to detect all the events are the key for improving our results in the IVF lab. Several clinical trials were published, and in 2017, this meta-analysis concluded that using time-lapse technology for embryo selection could improve pregnancy and live birth chances. But you know that there are also other publications that concluded that it was still unclear and that more trials were needed. So one of the great interests of time-lapse systems is the possibility to annotate each step of the development and to analyze their timings. That is what we call morphokinetics. This annotation is still manually performed, and it can be really difficult for the embryologists. They have to be well trained and to perform external quality control and also internal quality control really frequently. So we have to annotate the other stages that were described by Siré and colleagues. And it can be really difficult, especially when you have a lot of fragmentation, for example. It is also really well described that a huge variability exists in the timings of annotation between the embryologists in the same clinic and also between clinics. And on this representation from a paper from Martinez, Granados and colleagues, who studied all the results from the Spanish External Quality Control Program for morphokinetics. We can see that there is a huge variations in the timings annotated for each stage. So these results are really interesting, and they help us to understand why it is still so difficult to compare the results between the clinics. So many algorithms were published using morphokinetics in order to be able to select the embryo with the best chance of giving pregnancy. And these algorithms were designed to be used and work in the clinics that were developed. But you can see on this graph that when you try to apply these published algorithms on your own population, so this is the population of the clinic I work in, it does not work, and the scores given by the algorithms are not really correlated with the embryo implantation. So we can ask ourselves why, and I think that it can be explained by two major things. The first one is different populations between the clinics, and the second one, the difference in the way for embryo annotation. We have also to deal with different time-lapse systems. So that means different type of images, different type of softwares for embryo annotation, and sometimes different culture conditions. So what do we need now for proving the interest of time-lapse? We need large multicentric clinical trials, combining different kind of time-lapse systems and more reproducibility in the way to annotate. And we think that automation of embryo annotation is a key for answering this question. So the group I work with developed software that is able to automatically annotate embryo development. It is based on image analysis of a zone of interest, analysis of the variation of the GWAS levels inside of this zone, and also the zona pellucida detection. It can also read automatically the time of development and the name of the patients. So we drew on a pilot study, which was published by Mulder and colleagues in 2015 to develop our technique. And so the inside of the zona pellucida is automatically detected by our script, and then it focuses only in the study of the variation of the GWAS levels inside this zone. So on the image on your left, you can see an oocyte, and you can see that inside the zone of interest, the variation in the GWAS levels is small. But on the other hand, with this 88 stage embryo, we can see that there is a lot more of variation in the GWAS levels. And so by following the variation of these GWAS levels all along the embryo development, we can establish a curve of variation which is typical and really reproducible between embryos. And you can see that each time you have a cell division, a lot of variation occurs, and you can detect automatically the different stages. For the expanded blastocyst stage, we don't have any information anymore with the gray levels because of the expansion. And on this curve, you can see the automated annotation marked in red and the manual annotation, which was performed by the embryologist, is in green. And we have quite a good match between the two annotations for this embryo. Here you can see two curves of variation of gray levels for two different embryos. And you can see that despite the differences in their timings and levels of variation, they have the same global shape. And that is what is the most important to annotate. So I told you expansion results in the lack of signal when studying the gray levels. So we had to focus on the zona pellucida to be able to annotate TEB, so the extended expansion blastocyst. So we have an automatic detection on the outer and the inner zona pellucida, and we can automatically measure the distance between those two lines. And when it's half the original, it annotates TEB. So these are our results. We tested our software with more than 700 embryos, and we can observe a nice global correlation between the software and the embryologists for all the stages. Then for each stage, we applied the coefficient of variation that was described in the results of the Spanish external quality program to define acceptable ranges of difference between the two annotations. So this is the gray lines on the graph. And you can see that for the first steps of development and the last stages, we have quite a good correlations between the manual annotation and the software. But our software still suffers from limitations. So we only designed it for a research purpose to be able to annotate a huge amount of videos with a good reproducibility. But for a clinical practice, we miss the number of pronucleus and moreover the blastocyst morphology, which is still really important for embryo selection. So we think that artificial intelligence is needed in addition of this kind of technology. So this is image analysis to be able to grade the blastocyst. And today, human is still necessary. So for conclusion, I think that automation makes possible to annotate hundreds of videos really quickly, to annotate videos from different devices and with a perfect reproducibility. And all of that is needed to highlight new markers of embryo capacity, but it is still not enough for clinical practice, and human is still necessary. And so AI is the next step for that. So I wanted to thank all my colleagues from the IVF Center, and especially Magali Fayeux, who performed the image analysis. Thank you for your attention, and if you have questions.
Thank you very much, Arnaud, for that very nice talk, and which comes from a very clear and concise paper. We've had a lot of questions about time-lapse annotations and time-lapse systems in particular, but we've just got time for one question at the moment, and going with the theme, do you think we'll ever be able to trust completely what we're getting for these annotations and these predictions?
So I see that the human eye is still necessary because there are sometimes some events that you want to perfectly detect, not to transfer an embryo which has no chance to implant, like the number of pronuclears, direct cleavages, those kind of things. And I think that we can trust the machine, but still human has to validate the results before transferring the embryo.
Okay, and just while our next speaker, Amir Arav, shares his screen, just one quick question from who's sent. What, in your opinion, is the most important time-lapse parameter as far as embryo quality and prediction for the best embryo? Could you give us, could I put you on the spot on that one?
This is really debated. So it has been shown that the first divisions were the most important, but in our lab, we think also that the speed for distillation is really, really important.
Right, okay. Thank you very much. And sorry for putting you on the spot. Our next speaker is Dr. Amir Arav. He has many innovations, but he'll today be talking about a simple and automatic procedure of IVF and vitrification. So, welcome, Amir.
Okay, so can you hear me? Good.
Loud and clear.
Good. So, hello everybody. I'm going to speak about automation of vitrification and also IVF. And Jacques Cohen asked me to answer the question if what's going to be in 30 years from now. And to answer this, I went back to see what's happened 30 years ago. And this is what I did 30 years ago. I developed a technique for vitrification. It's called the minimum drop size, where you place oocyte on a transparent carrier, cryocarrier, and with a very small volume of less than 0.1 microliter. We plunge it directly into liquid nitrogen or liquid air, and we had a very good results. So this was in '92, and I remember very well that when I presented this, and this became the method that everybody's using today with cryotop, cryotech, cryolock, vitrifit, and so on. So I remember when I presented this meeting, Alan Transon came to me and he said, "Oh, it's an excellent method, but probably in the right hand." And he was quite right about this prediction, because there's a lot of variable between labs and also within the lab, as we can see here by this paper that came last year in Jacques of the group of SL Tan, showing that with sibling oocytes, there is a big difference between the results of senior embryologists and junior embryologists, something between 70 to 16% only. So we need standardization, and we need it to be done with automation. And so we said, okay, so if we want to do automation of vitrification, what do we need? What do we need to have? This is the first thing that everybody, when they do automation, have to be clear about. So with vitrification, we should have a very high cooling rate and warming rate, because we are dealing with chilling sensitive cells like oocytes that need to be very rapidly cooled and warmed. The volume, as I just said, have to be very small, less than 0.1 microliter, to enable to reduce the cryoprotectant, the concentration of cryoprotectant. It should be easy to operate, and it should be easy to store in liquid nitrogen. We don't have a lot of space in liquid nitrogen, so it has to be small, like the cryocarriers, for example, like most of the cryocarriers. It should be all automated, not by steps, not like only doing the cryoprotectant procedure and then to do it manually. It doesn't have any sense that you have stations, you have breaks. And also the warming and the dilution, everything has to be automated. We should take care that we have a very sensitive cells at this stage. So we should reduce mechanical damage. And everything had to be short time, which means that all the preparation for the next patient have to be very fast, and everything have to be in a short time. And it should operate in open system, which is needed for all sites, for example, and also for closed system. So 8 years ago, we started this project, and it was quite complicated. We produced something like 22 prototypes. I can tell you that we tried almost everything possible, and some of them were very complicated. And in the end of it, we came out to a very simple solution, which is a small grid, or that you can see here on the left, which have about 1 millimeter of diameter and has a pores, square pores of 50 micron each. So here is the oocytes, and here is the pores. And this is sitting in the tip of the straw, regular quarter cc straw. So you first load the oocytes or the embryos in the straw, and then you plug it just like that, and just plunge it in, and it's inserted already. And now you can move through the solution with the idea that everything, so this is the capsule and this is the straw. So everything will, all the solution from one solution to the other will penetrate into the straw by capillarity, and the embryos or the oocytes will sink down to the filter level by gravity. So here it is, and it's worked very nicely, both manual or with a device. So the device, robotic device called Sarah, and this is my daughter, Ayala. She's not Sarah, she's Ayala. And she did the first oocyte vitrification just by pushing the button. So what's inside Sarah? We have 6 straws, mini straws for oocytes and embryos. And we can have also large straws for tissue. So we did also testicular tissue and ovarian tissue using this type of straws. So we first load, what is it? Sorry. Let's see the bottom here. Okay. So we first select the time and the procedure that we want to do. It's different between cells, oocytes, embryos, tissue, and so on. And then the straws will start to move from one one station to the other in the predetermined time. And again, the solution will go in by capillarity. I'll jump to the end just to show you that in the end of it here, the solution will drain by absorbing paper, and the straws will go up and move in about 30 seconds into the liquid nitrogen or the liquid air, which is sterile in our case. So once it goes in, it will stay there and wait to the embryologist. So we still need embryologists here to go and take it and put it in the tank, okay, in the right place in the tank. So this is still manual part, but the whole, now it will stay there in the liquid nitrogen until the next step. So results, we published this on Jacques 2 years ago. With mice, we had a very high survival between oocytes and acyl stage and blastocyst stage embryos. They all survived and developed. And bovine oocytes, which is more better model for human, as it is very chain sensitive. There was also oocytes and zygotes were similar results to fresh control. But I think the most interesting study was done by a colleague of mine, Sergio Leda in Sardinia, and he was in sabbatical in Australia. And he did a very nice experiment comparing two-step procedure to 6 steps, which is allowed by the device, all inside the stove without taking it out from the stove, so one step after the other. And the results were like that. Day 7 embryo was better than day 6. This was expected. But what was interesting to see was that the multi-step, the 6 steps of exposure was for blastocyst was better than the 2 steps. And this is, 24 hours after the warming, which means that it's also including hatching rate. So he stained for apoptotic cells, which are in green here, and you can see that the 2 steps produce a lot of apoptotic cells, and the dynamic, the 6 steps, was they had almost no apoptotic cells, so very compared to a control. So in the veterinary field, it would be very important to use this idea because we have something that's called direct transfer, where a farmer can take the embryos, frozen embryos, from the tank and transfer them after warming directly into the uterus. But it's not possible to do it with vitrification because you need a different step of dilution. And this is why now, if you can do it inside the straw without to take it out from the straw, you can easily do direct transfer with vitrified embryos. And another example is this guy. It's a friend of mine. His name is Amos here in Israel, and he's a horse vet. And he doesn't have any idea of how to do embryo manipulation, but he's now doing a vitrification of embryos quite easily. Because he doesn't have to take it out from the straw, and after warming, he just take a look at them inside the straw, under the microscope, see the expansion of the embryo, and then transfer it to the uterus. So maybe it would be possible to do it in human, in the clinic. So the doctor will have liquid nitrogen with blastocyst, and he will warm up the straw, put them in different solution, automatically with the SARA or manually, and then watch it with a microscope, culture it inside the straw for 2 hours, then adapt it to embryo transfer catheter and transfer it. Now, very recently, we produced more results with sperm separation using the capsule. And we found out that we can do in vitro fertilization inside the straw just by plunging the straw with the oocytes inside the raw semen or frozen thawed semen. So if we have this part ready with culture inside the straw, as I said before. So now we are ready for IVF, in vitro culture, vitrification, warming, embryo transfer, everything inside the straw without to take it out from the straw, manually or in the device SARA, both. So we did also tissue. This is my last 2 slides. We did the testicular tissue using vitrification with the EVIT, with the straw. And these are the results. You can see it in our publication on JAR. And also we did human ovarian tissue with the EVIT, and also show that there was no difference between regular vitrification and our EVIT vitrification. Now I want to put your attention here, just to take this opportunity to tell you that we should be very careful about ovarian tissue and testicular tissue because they have the ACE2 receptor for COVID-19. And this can be very dangerous if we use liquid nitrogen, which contain the virus, because it's very easy to just vitrify virus because their size and the drop size of the aerosol. So here we can offer a device, a very small benchtop device, that make liquid air, sterile liquid air, in about 10 minutes. It's produced about 200 milliliter liquid air, and you can use it for your vitrification safely without the risk of contamination. Okay, I want to end up with other in the market. There is Gavi from Genea that do the preparation of the cryoprotectant before the loading that you have to do manually, I guess. And ESCO promised that in the next year they will have the first prototype for automatic vitrification. Okay, so summary. IVIT is a quarter cc straw with 1 millimeter of capsule. It can vitrify oocyte, embryos, tissue. It's operating safe because it's a liquid air. Open system and semi-closed system. Cooling rate and warming rate is very rapid, about 20,000 degrees per minute. We can vitrify 30 oocytes simultaneously in 17 minutes, or 6 blastocysts in the same time. And it's automatic insertion into liquid nitrogen, and also can be automatic warming and washing and culture of the oocytes or embryos. So I just want to thank people working with me, Pasquale Patrizio from Yale, Stoyana from New Hope, New York, Pablo Levisetti in the Humanitas, Milton Leong in the Hong Kong, Lisa from the Roche Lab IVF here in Israel, and my group, Fertile Safe in Israel. Thank you.
Thank you very much, Amir, for that excellent talk. You're on dangerous ground now. You're talking about if clinicians can actually do the transfers themselves. We've had a lot of questions. Thanks for the triage team. We've just got time for one question. It's by Barry Baird. How do you prevent cross-contamination in your system, SADA, between consecutive patients?
Good question. We do one patient, so the solution is can you hear me? The solution is one solution for one patient. So there will be difference between, of course, between solutions and patient, and there will be no cross-contamination, of course.
Okay, and a question which many people have asked, is it for any vit medium, any brand? Can we use that?
Any
type of manufacturer, for any type
for the solution. Right now, we are working with our solution, but it's not something very special. It's just we are doing it ourselves, and all the groups that are working with us, they do it themselves. But it's a simple solution that probably very similar to the others.
Okay, thank you very much. Last but certainly not least, I'd like to ask to Anna Garcia Poyo to talk about her innovation, which is the robotic embryology lab in a tiny box. So welcome, and over to you, Anna.
Hello, everybody. Thanks. I really feel honored to be representing my company, Virtual Life, here in this initiative. While I was obtaining my engineering degree at the Technische Universität of Darmstadt in Germany, one of the most demanding engineering schools, I realized that any technical problem could be solved if one worked hard enough on it. And that's because engineering is based on equations and principles that tend to be predictable. In the biology world, we cannot apply the same principles, and that's why complexity increases. It was my father who told me the beauty of chemistry and physics, but I was also fortunate to have an ancient Greek professor in my family, my mother, who taught me not to forget humanities. And that's why today my goal is not just to speak about the technical features of the medical device we are developing in our company and that I'm very proud to show today, but also try to emphasize the value we are creating with IVF automation. When I mean value, it really implies all the stakeholders of the industry, from embryologists, IVF directors, IVF centers, and never forget IVF patients. I have been very, very fortunate also in my career. So I am proud to say that I got a CEO role when I was 32 years old, and the pregnancy never caused for discrimination, but I know that's not usually the case. And that's why I will start my presentation showing how the industry, IVF industry, looks like today. I think it's very well known that new social lifestyles, new demographics, our generation are having babies 10 years later than our parents did. On the other side, many women still need to decide whether they go for work or family. And I think that's where over to Saaj comes in, that have healthy babies when you're ready and democratize through technology. I think that's so important that we focus on egg freezing on one side, IVF ICSI, and that's why it's so important. So 40 years ago, IVF, the first kid from IVF was born. I was not working for IVF at that time. I was almost born. But if you look at the labs and you make a picture today, it's incredible to see that we have seen some process optimization. Media has been changing extremely. Also, we have new technologies like optics, incubators, new microreproductors technologies. But looking at the hardware itself and how IVF is taking place in the laboratory is still a very uncontrolled environment, and that's where we think hardware or a company like ours could provide embryologists with the right hardware, and yeah, because in the end, highly skilled embryologists are used for very repetitive tasks and consuming, and it's really expensive to equip those labs and to growth. There is a last point that I would like to highlight also in terms of the IVF industry. I took the example of the United States IVF industry. Thanks to a study we did with the company MedAnswers Fertility Answers, that's an app platform from infertile or infertility aware patients. And what we did here, we calculated the distribution and the distance users had to the closest IVF center. So in red, what you can see is the distribution of IVF centers. In blue, with different densities, you can see the patient's density. It was amazing to see that there were patients that had to drive 4 hours to go to the closest IVF center and receive human reproductive treatments. On the other side, between 20% and 50% could really benefit from what it's called a satellite clinic. And I think that's where we also create a lot of value, because having a tiny IVF lab in a box, then we're reducing barriers for entry for small clinics, and at the same time, we are reducing the footprint to grow in areas, in remote locations where patients also need to receive IVF. I will build the product presentation starting from one technology that it has been a success, and it's the great example that technology and automation can truly assist embryologists in their day-to-day work. And that's the time-lapse incubator. So it was the first time it was automatically taking images from embryos during the different events in the embryo culture. The PN visualization provided flexibility. Embryologists could assess what happened in certain events during the weekend without having to go to the clinic in the weekend, and the final decision was taken by the final embryologist. So building on this specific product, I would like to say that there has been some automation in specific areas in the workflow of IVF. So today, Amir showed a prototype also on the vitrification preparation area. There has been embryoscope and other embryo culture, and we really believe that it makes sense to avoid all these steps, avoid the embryo disturbance within all these steps, and in Over to Life, we believe in integration. Very proud to share the product that we are currently developing. It's called the Natural Life. So the Natural Life, it's an embryo making and embryo freezing machine. So the input will be oxides and sperm. We will fill up with all the different reagents. 5 days later, we will have embryos genetically tested and ready to freeze. I know it seems like really something's going to happen in 30 years from now, but we really advocate that it's going to be faster. So what are we going to provide with this machine? We will lower the process cost, we will improve consistency, we will continuously monitor in the same way the time-lapse was doing for the culture phase. We can monitor the different phases. So from fertilization, denudation, sperm wash, embryo culture, non-invasive embryo testing. I will explain a little in detail afterwards, and the embryo vitrification. Our product consists of 2 different parts. So on one side, we have the biotip. Everything is happening in one single biotip. This will be 1 per patient. We have the capacity for 12 embryos, and the different processes, they will be all integrated. So IVF is going to happen in a chip. On the other side, the Natural Life machine will be the fluid control unit, the temperature gas control, and the time-lapse. This biochip, and I hope many embryologists that will be joining right now, they will understand what we are trying to do with that. So because the current manual process implies using 76 different consumables. So our biochip will replace in an efficient way all these 76 biochips. We will have optics. Yes, we will have optics for doing the time-lapse. We have developed our own optics system. These are actual pictures taken by our system. And I think what we are trying to get is the quality is comparable to other systems that there are already in the market, but the footprint and the size is much smaller. And now the secret sauce that I haven't taken. Today, some other colleagues talk about soft robots, AI, and here we want to talk about microfluidics. So our machine is a microfluidics platform. What are we trying to do with microfluidics? Microfluidics, we are trying in the end to mimic what is happening inside the body. So we believe that's the ultimate in embryo protection, because when the embryo is not going to move, it's going to be always in a safe way. It's not going to be disturbed. We have calculated, and in the manual process, embryos are disturbed up to 70 times. And with our system, there will be maximum 4 times. So with all this together, in a very controlled, small space, we believe the success rates will increase, and we will reach the enough consistency to compare patients with comparable parameters. There is a last point related to integration, and is that today we discussed different technologies related to embryo selection. We know that there is AI computer algorithm. We know that there is metabolomics testing. That's a very specific proprietary testing that we have developed also in our company, and non-invasive PGT. So the aim is to have all 3 techniques in one device integrated. So I am very proud to share today at Over to Life, we have our own non-invasive metabolomic testing. We are able to rank embryos according to in a non-invasive way. We analyze the sperm media of the embryos, and then we are able to rank them and say which ones are going to implant and which ones are not. We have carried out a study with more than 100 embryos, And we are, depending on the different algorithms, between 85% to 90% accuracy. And there is a free prospective study ongoing, a new one in the next month, so that for a general information as well. And it was developed, I forgot to say that, by our CSO, Jose Orcajada. So how our hardware and platform is going to improve that is because we are able to sample the non-invasive liquid that we need for testing in a repeatable way. We are going to minimize the contamination of the samples. So we really believe that with one machine of ours, it will improve in accuracy. Very, very thankful and very to all the team that you see here. So we are about 26 people in Madrid, different engineering disciplines, optics, mechanics, microfluidics, electronics, embryologists, research, bioinformatics, and there you can see as well a very outspoken and respected professionals from the IVF world that are our founders team. So our CEO is Martin Wasarski, the founder and CEO and chairman from Prelude Fertility, the fertility networking, clinic networking in the United States, Santiago Munet, who built the biggest embryo testing company in the United States, and Jose Orcajada, our CSO, who was also the CSO of Ivy Foundation. Our investors, so we're a VC-backed company. Lead investors are Khosla Ventures, Felicis Venture, Rakuten, and some other personal investments. And our consulting embryologists are Gary Smith, Gloria Calderon, and Nuno from Embryo Tools in Spain as well. So I am going to share. I hope everybody is listening correctly.
Patient eggs are separated from the retrieved follicular fluid and transferred to the overture biochip. One egg will be placed in each well. IVF culture media and other reagents are loaded into the biochip reservoirs. Next, the biochip will be inserted into the cradle, which contains pumps for moving fluids around the biochip. Firstly, the cradle will prime and equilibrate the fluids in the biochip, and when ready, will proceed to push sperm samples to each of the wells containing the eggs. Overnight incubation will lead to fertilization of the patient's eggs. The fertilized zygote will then be cultured on the biochip for 5 days in controlled gas and temperature conditions. At this stage, the embryo culture media is sampled, collected in separate wells, and analyzed for non-invasive genetic and metabolomic screening. Once the process is complete, the embryos will be vitrified with a series of fluidic exchanges using cryoprotectants. As soon as these steps are complete and embryos are equilibrated, these are now ready for transfer and storage in liquid nitrogen. The biochip protocol is now complete.
Okay. So in this video, we show for the first time how our machine works. And to conclude with the presentation, I would like to give an answer to the questions. If at Roberto Live we believe there will be less embryologists in the future, we truly believe there will not be less embryologists in the future, because automation, what it's going to do is to increase consistency, but at the same time increase usage of ARTs, so assisted reproductive technologies. So there will be more embryologists needed, but we believe that maybe it's a good point in time to rethink how the role of the embryologist will look like. Probably there will be less pipetting steps like there are today. There will be more research, and there will be more coordination and probably a closer contact to patients. Thank you very much. That's the end of my presentation.
Thank you very much, Anna. That was excellent. Excellent. It has everything. It's got time-lapse, metabolomics, of course we know, and and microfluidics. Okay, so it's good that they're all coming together. There's been a lot of questions. One that points out, and it goes back to your last slide, is it's all about scale and cost, and all about, with all these technologies in this little box, has anyone done a cost benefit ratio to see? Because obviously what's involved in the cost for the production and that machine versus employing 3 embryologists, 4, or however, has that been done?
You mean in terms of capacity? What we have calculated, for instance, is that one embryologist, imagine that the ratio to embryologists is one embryologist can handle with good quality 100 cycles, right? Let's put a number. So with our machine, our estimate will be that we can do 300 to 400 cycles per embryologist.
And of course, it would have to depend on how much everything was and how much you paid the salaries. But
okay, in terms of savings, you mean? Well, yeah, I think what we can say is that we know, yeah, because that's a different, very difficult questions, because it will depend also on the country and how we are saying, but we know that with the same team, also a clinic can do 30% more of the cycles, even paying more the embryologists and having like 10% more benefits in the clinic. So what we are trying to make, and I know it's like the Tetris exercise, is to show that even paying more the embryologists, the clinic as a whole can really have a bigger throughput, and yeah. So we make the joke that the embryologists in the end, they will be happier with our machine, and it's probably right.
Okay, thank you very much. Jacques?
Yeah, thank you all for these wonderful lectures. There are a lot of questions, starting with Amir. Dean Moorbeck is asking, are there published studies on volume on cryocarrier for oocyte vitrification, for instance, comparing less than 0.1 microliter versus 0.2 microliter? Are there differences there in these small volumes when you go up a little bit, 50% up, 100% up? Do you know if there are actually differences?
Between 0.1 and 0.2 microliter.
Yeah.
We didn't look at that difference, but we did a lot of study between 0.5 and 0.1 or less than 0.1, and there was a big difference in terms of nucleation and of term of crystallization. But the nucleation, ice nucleation, is depend very much on the volume, and when you reduce it, you just reduce the chance that ice nucleation will appear, and this is the most important actually.
Question again for Amir. Samar Farhat is asking, how many embryos in the cryo-leaf in Sarah can we put? So how many embryos can you freeze at the same time? And how long is that cycle? How long can you change the whole thing out and go to the next patient?
Yes, the whole cycle together with the preparation will take about 15 to 17 minutes. And we calculate that in each straw you can put 5 oocytes. Although when we did it with mice, we did much more than that. But in human or with bovine, we do it with 5 oocytes. So it's 30 oocytes altogether for one patient. I think that this will be more than enough. When you do blastocyst, you'll have to do it 1 per straw, because you want to warm up only one blastocyst at a time. So it's 6 blastocysts for 6 straws. This is what we do. When you do another patient, it will be after 17 minutes.
Right. Thank you very much. Anna, when is this thing ready? Somebody's asking here. I don't have a name who's asked this question, but when is this?
Yeah.
When are you going to launch this?
Okay, so I guess everybody, or maybe I can describe how the regulatory process looks like. Right now we are in the preclinical phase, so we can say that in the next months we will make results available. And then we have to enter the validation phase and the clinical phase. So I would estimate we can test it with human 1 year from now, 1.5 years, but that doesn't mean that we can really start selling. So in the clinical phase.
Okay.
So probably in the market, yeah, so between 1.5 years and 2 years.
Okay.
Great. And can I pose a question to Minnie? Prof. Minnie Saaj. A lot of questions about your robotics. It's soft robotics, and I can see where the question's going. Could you be envisaged that you could use it at very low temperatures, like in liquid nitrogen, perhaps, which is, you know, minus 109.95 or 96? So can you use that in liquid nitrogen?
Well, as of now, I cannot say, or I cannot give a definite answer, but the whole aim of designing soft robots for clinical application is to make sure that it's a biocompatible material that we use. And therefore, depending upon whether the soft robot we are using for fertility treatment is for in vivo or ex vivo application, accordingly, we can change the material. So for example, for in vivo, we use the silicone, which is biocompatible. Whereas if it is for external manipulation purpose, we can very well use other materials, which can be compatible for use with, say, for example, extremely low temperatures. So it depends where we are actually using this instrument.
Okay, right. Thank you very much. And a question for Arnaud. Your annotation for the time-lapse, it can be used from clinic to clinic. There has been some validation done, yes, on other clinics.
No, actually, it can be used on any system, and it's really independent from your clinic or the way you annotate. It's you can use wherever you want.
Great. Okay. Okay. Thank you very much. And for Anna, do you think that having IVF in a box will help the appeal for IVF in more remote areas or in less developed countries?
Yeah, I think it's a very good question. So when we studied also globally the IVF industry, we realized that probably the needs in the US are not the same that the needs in Europe and are not the same needs that you have in the developed countries. It is well known that in countries like India and in Asia, there is lack of infrastructure. So in those cases, we really believe that having something very small will help increase this infrastructure. On the other areas, as I was showing in the United States, it's very well known that we have a lot of competition of clinics in the biggest cities, but there are people that have to travel, and I don't think that I think it's very hard to have to travel for some hours, the IVF journey, the psychological aspect. So everything that you can have closer, it will help up, affordability, and it will increase democratization. That's what we want.
Okay, thank you very much.
Anna, I'll just stay with you, but before I ask you the question, maybe Thomas, maybe you can bring up the two closing questions. We still have about 5-6 minutes, and then the two closing questions are: question one, after listening to these talks, I think robots, AI, automation taking over a reproductive scientist's job is more likely, less likely, or about the same? And the second question is, in your opinion, which procedure will be the last to be automated or robot assisted? And the list here is egg retrieval by the clinician, egg collection by the embryologist, semen prep, ICSI, vitrification, embryo selection for transfer, embryo transfer. So if you could quickly submit these two questions for the poll attendees, that'd be marvelous. Anna. A question from Deepti Tiwari, who is asking, do you have a backup plan in case of power failure, software malfunction, and false alarms? This could be a question for all of you, of course. I mean, Minnie, I mean, you must be, every robot you've probably produced and helped to engineer. We'll come back to you, Minnie, because we would like to know what's your backup plan when things go wrong?
Yeah, I think I would answer this question also to the point that I was mentioning before with validation, right? So we can demonstrate that our system now works with animals. That's I think a very good assumption of it. But then when you go to the clinical phase, the FDA, the CE mark, they want to see for every risk that you have in your machine, a contingency plan in terms of, and also assess the classification of risks. I hope if our regulatory manager is looking at me, I'm not saying something wrong, but I think that's how we should develop the machine, and that's why it takes so. But in our case, I think the fact that we have everything in one single place, all very controlled, and we are monitoring everything, is that we can have evidences of what's happening all the time.
So what you have doors there, these things can fail. I mean, glove compartment doors in cars fail, and you have to, do something about it, which usually means pushing and pulling. You can't do that with this unit. So you must have systems in place before you go to market. How does this work in general before you go to production of, for instance, a medical robot? You have to be absolutely sure that all of these accidents and issues like power failure are taken care of, right?
Yes, a very important part of design engineering any product, whether it's a medical robot or any other platform, is to really look at the risk analysis, which takes into account not only the technical faults, but also the operational side of things. So when you design a product, we do look at, system or subsystem level diagnosis, health in diagnosis. And this actually gives us, a big list of potential failures, and we can look for mitigating those risks. So these are all part of the different phases of this design exercise. And I believe, we will get the approval from FDA and other agencies only if we can present a robust system.
Yeah, regulators would want to know all these things. Amir, when your machine jams, you must have ways to get around it, right?
Yes, I generally don't trust very much machines, and and this why what I had in mind from the beginning was that every step that the machine does, you should do it also manually. So if you can do it manually, you can stop at each step of the machine and continues manually, so that you don't lose valuable embryos or oocytes. So this is one of the things that I was thinking about right in the beginning.
Right. We have the poll results.
Let's have a look.
Okay.
So here's the final poll we have here. After listening to these talks, and the question was, I think robots, AI, automation, different things, of course, is going to take over reproductive scientists' job. More likely, 39. Less likely, 22. You pessimists. And about the same, 39. Okay. Well, for those of you who are pessimistic or on the fence, there's another series of similar topics that we'll be bringing up pretty soon on this event as well. Second question, in your opinion, which procedure will be the last to be automated? What have we got? Egg retrieval by the clinician, 24%. Egg collection, 16. Semen prep, 3. So that looks to be ready to be automized. ICSI, 21. Vitrification, well, I think we've sold you on the idea that it can be done. Only 5% think that would be the last. And embryo selection transfer, 10%. That's a good number. So we're well on the way. And embryo transfer, 21%. So really, we're looking at the clinician side, who seem to be still in with the job at the moment. So over to you, Jacques.
Yeah, thank you. So a final set of questions. We're we've already run over time by 6-7 minutes, so maybe a minute. For Anna, what do you do about gas supply?
Gas supply. So we have decided so you mean if we have premix or we have our own mixing system, right? Yeah. That's a good question. So in the current prototype, we have both because we realize, as also was what Amir was mentioning before, maybe if you are in a big hospital, you have the possibility and you cannot control always the quality of the gas. On the other side, there are other clinics that work with premix. As for now, we keep the flexibility to that, but very like we are having both. But I would say that in the end, we don't want to skip this possibility to mix the gas ourselves and guarantee the quality.
Right, right. And so you will use any media. You don't suggest a particular medium.
Well, there is maybe one thing that is true is that for vitrification media, we do have an agreement with Kitasato. So we have Kitasato media, but for the rest is universal. So it will work with every media. The only process that we are is the Kitasato for vitrification.
Yeah, yeah. Amir, the same question was for you. Do you need any media? Or do you prefer a certain media? For Sarah? We can't hear you.
As I said before, we have our own media for vitrification of oocytes, different for embryos and also for tissue, use different medium. But these are something that every company can build and do for us, because for example, we use many steps for the exposure that we found that it's better for all sites. Yeah. So and this is not provided by any company right now. So you have to build it yourself.
Thank you. And Arnaud, actually the same question for you. What happens to the annotation system where you change conditions? So when you make the volume higher, for instance, a little bit higher, would that make changes? I mean, you have to test it out, I guess, right? I mean, it's a question.
Yes, well, normally, as long as you have the image, you can annotate whatever the kind of image. So I don't think that it would impact the way for embryo annotation. Okay. We know that different medias or things like that doesn't change anything.
Right, right. So what's the plan to get blasticists involved in annotation? What do you think is needed to do that?
For the blastocyst morphology?
Yeah.
Yeah. We have to use a different kind of technology. It's going to be machine learning. Okay. Because the difficulty is that we have only one plane of the image. And if the initial mass is not in the middle, we can't, we can't evaluate it. So with machine learning, we would be maybe able to have more information.
All right, panel, I think you've done really, really, really well. Great talks and excellent answers. Giles, I think we need to wrap this up. We've gone through our program. Thank you, audience, for staying with us so long. I'd like to thank a whole host of people. There were 16 people involved in this meeting. First of all, my co-moderator, Giles Palmer, the speakers, of course, the background panelists, Colin and Kelly, the Q&A, Kelly and Marlene, the Q&A triage, Shaista, Alison, Liesel, Peter, and Dara, and then the logistics, getting the Zoom to work properly. Thomas, Elliott, also thank you, Thomas, for that introduction video, and of course, Marianne. I wouldn't want to close and not announce the next session, which will be on the coming Friday this week, same time, same place. It's called Rebuilding Ovarian Function. And it's brought to you by the University of Colorado Department of OBGYN. That's Friday, 24th of April. And two speakers are Professor Dror Merov and Christiani Amorim. And it will be organized by David Russell from Colorado, Cassandra Roeka, and Leslie Appiah. So we'll see you back on Friday. Please register for this. And again, thank you everybody for this. And you at home, please stay safe and see you on Friday. Take care.